Welcome back to Click for IB Biology, where we're looking at ventilation. Now, ventilation, of course, is the process which allows us to maintain the concentration gradient of oxygen and carbon dioxide across the alveoli capillary interface. So we'll take a look at the ventilation mechanisms, which include quiet breathing and maximal breathing, and ventilation patterns, and identify these from graphs. Now, should you want to, you can download a PDF from the description of this presentation, which will allow you to create your own annotations and to check your understanding by taking the quiz at the end. There you'll also find links to the next part of the syllabus. Okay, so the principles of diffusion, as we have discussed before, are all about increasing the surface area to volume ratio. And we find that size is an important feature, the tiny sizes of the alveoli. The fact that we have this folding of these membranes inward increases the surface area. The flattening of the alveoli pneumocys 1 creates short diffusion distances. But now we need to consider how the gradients are created and maintained. So within the lung, we have a unidirectional flow, inhaling and exhaling. The inhaling brings in atmospheric oxygen to create a high concentration in the alveoli relative to the blood supply, so we have a diffusion gradient this way. And then at the same time, carbon dioxide will diffuse into the low carbon dioxide in the alveoli, and then that would be exhaled to maintain this gradient. So inhaling and exhaling are responsible for establishing the concentration gradients. Now, considering how does inhaling and exhaling actually occur, let's think of the ribs with the sternum at the front of the chest here, at the front of the thorax. And we have the ribs, 12 pairs of ribs, in which pairs 11 and 12 are not connected, but are known as the floating ribs. Ribs 1 through 7 connect through cartilage to the sternum, and then 8, 9, and 10 connect to the pair 7 above. These connect at the back with a gliding synovial joint to the backbone. Now, how do we get the rib cage to move? Well, the answer to that is, of course, the muscles. So that creates a lever system with the rib cage. And we have the external intercostal muscles, which are used with inhaling. And these lift the rib cage up and outward, whereas the internal intercostal muscles, they would pull the rib cage down and inward, which we associate with exhaling. Additionally, we need to consider this structure here, which is the diaphragm. And the diaphragm we can think of as a muscle separating the thorax from the abdomen, taking this curving structure like so. And when in this position, this is consistent with when we have relaxed or exhaled. But when we breathe in, the muscle, this taken by the red line here, will shorten to a straighter structure like this, and that we would call, that's when we are inhaling. The kind of breathing that you're doing as you are relaxed are called tidal volumes or quiet breathing. But where one uses one's rib cage and you're trying to inhale large amounts of oxygen and air and to exhale, such as during exercise, now we would be working towards what is known as our vital capacity. So as we sit relaxed, this is what happens when we inhale. Our diaphragm contracts and it flattens. This causes a stretching of the lung tissue, and so the alveoli are stretched and their volume increases, as a consequence of which the pressure inside the alveoli decreases. So now air flows from outside of the lung at higher pressure to the lower pressure inside the lung, and we have inhaled our tidal volume. It is possible to add to this process a contraction of the external intercostal muscles, pushing the rib cage up and outward, and this further stretches our lung tissue, further increases lung volume, and reduces the pressure with a greater inflow of air into the lung. Exhaling, on the other hand, requires a relaxation of the diaphragm. Lung tissue contains elastin fibers. Think of these like protein rubber bands. So that when the diaphragm relaxes, these elastin tissues recoil, compressing the lung structure, reducing the volume of the alveoli, increasing the pressure within the alveoli, forcing the air out of the lung. Again, this can be enhanced through the addition of the internal intercostal muscles, which will pull our rib cage down and inward, heightening the compression of the lung tissue. Now, interestingly, one can also contract one's abdominal muscles at this time, forcing the abdominal muscles organs inwards and upwards, giving a further compression to the lung tissue. These gas exchanges can be measured using a structure called a spirometer. In this illustration, we have the classic bellows spirometer. The mouthpiece is here, the bellows are here, and this structure can move up and down with inhaling and exhaling, and this can be recorded by a data logger or a kymograph. Soda lime is present to absorb carbon dioxide, so that we're not rebreathing carbon dioxide which has been exhaled. The kind of trace which is obtained by either method looks something like this, where this volume here is the inhaling and exhaling, known as the tidal volumes, which is approximately half a litre or 500 milliliters. If you ask a person to inhale as much as possible, the trace would go to this level. So now we're using our external intercostal muscles. And then to exhale as much as possible, that would take the reading down to this level here. And the distance between the two is known here as the vital capacity. Now the size of the vital capacity depends upon how big the person is, but for an adult, we're looking at somewhere between 3 to 5 litres. Let's take a look at some figures for the changes in gas composition with inhaling and exhaling air. Notice that there's very little change in the nitrogen. Notice that the water content, due to the moisture within the lungs, is a variable part, depending upon the atmospheric conditions. Of the approximately 21% of inhaled air, which is oxygen, approximately 4 to 5% of that is removed for gas exchange. Carbon dioxide, on the other hand, in inhaled air is about 0.04% approximately, and this increases to 4%, which is an increase of 100 times. For consideration, remember that there is no gas exchange in the trachea or the bronchi or the bronchioles. This is referred to as anatomical dead airspace, but probably only accounts for about 150 milliliters of air itself. Another component we should consider is that the alveoli are never entirely emptied. So if we illustrate an alveoli here like this, they always contain air, and this is called residual volume. Now, the purpose of the residual volume is to prevent the collapse of the alveoli. A collapse of the alveoli would mean the moist surfaces would touch, and it would be very, very difficult to inhale those, to inflate those again rather, without medical assistance. The next video in this sequence is to make a comparison between human gas exchange and that of fish in fish gills.